students, today we will discuss about the monocot embryo development, brief idea about seed and fruits as well as apomixis. Today, just you assume and look at on this picture that this is a zygote which is diploid structure, and as per earlier statement that the formation of endosperm takes place here, that is the PEN. So zygote will divide transversely to form two cells, the basal and terminal cell. Basal cell is projected towards the micropyle. This one is micropyle, means this structure is embryo sac, and the which one is the adjacent to micropyle, that is called as basal cell, and projected towards the PEN or endosperm, that is the terminal cell. Basal cells is transformed and then it becomes a vesicular cell like structure which forming a hausturial like structure which helps in the absorption of food from the endosperm, whereas the terminal cell divides by vertical division to forms basically two cells, and after that it will once again divided by the vertical division and forming this structure, and this is the vesicular cell which acts as hausturial cell, and helps in the absorption of food, whereas few tiers of the cells forming the suspensor, and just below the suspensor, that is the few cells which forming radicle, and below that few specialized cells which is termed as hypocotyle. Hypocotyle means this is the part of embryonal axis, and below that is the one side globular structure of the proembryo, that is the cotyledon, and another side which is forming the plumule, means these are the cells which form plumule in the future. And now this globular structure and few mass undifferentiated tissues turned as like this. In this structure, this is the longitudinal section of, this is the longitudinal section of well developed monocot embryo. In this picture, the single cotyledon, which is termed as scutellum, which is also referred as cotyledon, and its adjacent structure forming the apex is the coleoptile, and below the coleoptile, that is the shoot apex, means the plumule cells give rise to the shoot apex, and its covering is called as coleoptile, whereas just below at the bottom, this is called as radicle, and radicle is protected by root cap cells, and root cap cells is protected by coleorhiza, means the difference between coleoptile and coleorhiza is, coleoptile is the covering of shoot apex, whereas coleorhiza is the covering of radicle. Dear students, monocot means generally cereals plants, grasses family. This belongs to the monocots. Now we will discuss about the seed. But before description of the seed, I would like to explain about the fruit, because ovary which is fertilized to form fruit. So fruit is the fertilized ovary. Fruit has basically two parts, that is the pericarp and seed. This is seed, and again pericarp is divided into three parts. Epicarp, this structure, the outermost thick covering, it may be thin in case of tomato and brinjal, whereas this is the longitudinal section of the mango. In mango, that is the thick leathery epicarp is present. Below that, mesocarp, this fleshy part is the mesocarp, which is pulpy and juicy and mainly edible part, and below that endocarp, this covering, this line is endocarp. It may be thick or thin depending upon the species of the fruit. So fruit is basically this structure. Now, fruit may be divided into two parts, that is the true fruit and false fruit. I am not going to describe in detail, because in this chapter, in the NCERT, very brief idea, primary or elementary knowledge about the fruit. True fruit means those fruit directly develops from ovary, means fertilized ovary. Directly develops from fertilized ovary, like here, for example, mango, tomato, brinjal, etc. Whereas false fruit means the fruit is not developed from the fertilized ovary, not develops from fertilized ovary. For example, in case of apple and pear, the edible part in case of apple and pear is the swollen and fleshy, juicy thalamus. So this is the false fruit. Now I am coming on the seed. Actually, seed is the fertilized ovule. Fertilized ovule. And seed has mainly three parts. Seed coat, embryonal axis, and cotyledon. Seed coat may be single or double. Single coated seed having only testa, and suppose double coated seed having testa and tegmen. Testa is rough and tough, and it is the outermost layer, so it prevents seed or embryonal axis from the external factors. It prevents from the chemical action. It prevents from the high temperature and high humidity and other bacterial or action or fungal action. So testa is rough and tough, whereas tegmen may be or must be thin, filmy layer, and it covers the entire cotyledon and embryonal axis, and it prevents the loss of moisture of the seed. Actually, seed has 15% moisture. Seed has 15% moisture, and this moisture helps in the future for enzymatic action after absorption of water, because the seed which is kept in the any container in the kitchen and a store, that is the dormant seed, not dead seed. Embryonal axis having two parts mainly, radicle and plumule. Radicle develops as root, means taproot, that is the taproot, and plumule develops as shoot, that means shoot primordia, having leafy structure, that is the leaf primordia, and cotyledon makes the shape of the seed, and cotyledon has nutritious value which is used as food. Suppose in case of pulses, pulses is non-albuminous seeds having no protein layer, having no endosperm, but just reverse, pulses seed has abundant protein, and this protein is stored in the cotyledon. So cotyledon makes the shape of the seed. It may be spherical like pea, may be shield shape, in case of maize like this, like such type of seeds. In case of maize, the spherical, in case of pea, and bean, like bean, means a Dolichos love seed, that is the same, bean shaped. Just I want to say you that in case of cereals means major and minor cereals, wheat, rice, maize, jaw, jawar, bajra, in such cases seed is referred as fruit. So that's why in case of cereals, in case of caryopsis, that is the single seeded fruit, so this is the brief idea, and on the basis of a structure and on the basis of presence or absence of endosperm. Seed is of two types, albuminous and non-albuminous. Albuminous seeds are those seeds which retain some part of the endosperm, because the future of endosperm is that to help and to supply food in the development of embryo. That's why during the development of embryo, the entire endosperm may be consumed. Suppose few amount of or few cells of endosperm remain present within the seed. Such seed is called as albuminous. And cereal seeds are very prominent, whereas in case of non-albuminous seed, if the endosperm is totally consumed, and such type of seeds are the pulses seeds. Pulses, that means pea, gram, lenticels, etc. Kajan, that is the arhar. These are the non-albuminous seeds. One thing I would like to tell you, if some persistent nucleus may be present within the seed, and this nucleus, that is the persistent nucleus, which is also nutritious. In case of black pepper, coffee, beet, in case of nymphia, in case of cardamom, these seeds having the persistent nucleus. Friends, now I will talk about apomixis. This word was discovered by Winkler in 1908, and this word has two meanings. Apo means without, mixis means no marriage. Mixis means marriage. So the development of seeds without fertilization. So in case of apomixis, the seeds will develop by the process of asexual reproduction and no fertilization takes place, and the fruit which has such type of seeds may be termed as parthenocarpic fruit. Parthenocarpic fruit means the development of fruits without fertilization. In case of banana, that is very good example. In case of banana, and the another word is that parthenogenesis. Parthenogenesis means development of embryo without fertilization. Without fertilization, embryo will develop. So the both phenomena are related to apomixis, means here also without mating, without fertilization, development of seeds, and this type of seed is obtained by the process of asexual method, and such seeds or such plants which are performing apomixis is called as apomict, and the new offspring will be similar to the mother plant, means there is no change, means there is no any genetical change. So apomictic plants are genetically similar. So, when the two individuals are genetically similar, new offspring plants are 100% similar to the mother plant, means there is no variation in the genes. So, these genetically similar plants may be termed as clone or may be referred as, and we can treat it as clone, and the members of the such type of cloned animals, cloned plants are called as ramets. So ramet, clone, apomixis. We can obtain apomixis by apogamy method, by agamospermy method. It may be obtained from the diploid sporophytic cells, and which develops or treated as zygote. It may be nucleus. Suppose in this embryo sac, here is the zygote. Zygote develops as embryo, and the cells, these are nucleus. The cells behaves like diploid cells. These sporophytic cells, sporophytic cell behaves like diploid structure, and it is treated as, it behaves like zygote. So it will divide. When it will divide, it may form embryo. So within a single embryo sac, this is the natural embryo, this is the sexual means of embryo, and another here is the embryo. Suppose another cell may develop as embryo. Outside the nucleus, the cells of integument, it also behaves like diploid structure, diploid cell, and may behave like zygote. It also divide. So, and forming here also embryo. So within a single embryo sac, many embryos are present, and this condition is called as polyembryony. Meaning is that many embryos within a single embryo sac. This phenomena is generally visible in case of orange or citrus. Citrus genus has many species. Few species having only one embryo, but one species, Citrus revoluta, having 40 embryos within a single seed. So when we take the seed of lemon or any orange and we peel off seed coats, and we push many small structures like this is the seed of citrus seed. We just peel off, and when we will push, many small structures are come out. These are the embryos. So polyembryony takes place in the citrus. It has been seen that such type of event is beneficial for the Indian farmers and all farmers, because these seeds having more viability than the hybrid seed. There is one feature in the polyembryonic case, the seeds having more viable. So we can use such type of seed in one season, or maybe in alternative season in the future, whereas in case of hybrid seed, the farmers can use only the same year and they can obtain the good varieties. So this is the benefits of the polyembryonic. That's all.